I'm very happy and super excited again to be here in this forum and share some more insights on multi-omics, which is a rapidly evolving field and is having a massive impact at scale in pharma industry, but also in academic research. So I will share my screen, and then in next roughly 40 minutes, I plan to give you a bit of overview of the whole drug development process itself, like how the drug is developed when you think of a big pharmaceutical company. I will give you an overview of the omics technologies, which is very integral part of today's drug discovery programs from early phase to the late phase. And then what I would like to do is to give you a couple of examples from recent publications, which can give you flavor of how these technologies could be actually used to impact patient at scale and develop drug, mainly in the late phase of drug development. So, Gopal sir, I cannot see anyone now. So, if
Yes,
there are
yes, I
any
will convey
questions,
you. You are very
yeah,
much audible and your slide is also visible. Please
okay. So,
continue.
if there is any question, happy to take
Yes,
questions
yes.
while I'm talking, or at the end of this talk.
जी
So
इसको हाइड कर ली वो नीचे जो है उसको
before I
हाइड
dive, sorry.
इसको हाइड कर दे नीचे लाइन जो दिख रहा है ना शो प्रेजेंटेशन वाला वो लाइन हाँ यस यस प्लीज कंटिन्यू
Okay. Before I dive into the topic, there are two key elements which is important to understand before we learn about how that could be applicable in drug discovery pipeline. So we all know and have sort of like some sort of you can say prior knowledge that drugs are developed by these pharmaceutical companies or big mid-sized biotech companies or startup companies, and then eventually they get approved, and this is what we buy from the pharmacy. But I'm quite sure most of us do not really appreciate the fact that how much effort goes into the whole drug development pipeline and how much time it actually takes to identify a target and then to eventually develop a drug which can impact patient life. So, in one slide, I just want to give you an overview of the whole drug development process. What are the key steps? And then we will move on to multiomics. Any drug discovery program which happens anywhere, including academic setting, although it is, I will say, rare that there are drugs which are developed end to end in an academic setting, but there are some good examples where people are developing drugs within academia and then spinning out companies from academia. The very first step is the target discovery. Target discovery means that for any disease, first you need to understand what is the disease mechanism, how that disease is, what is the pathological condition, or what are the drivers of the disease. And then you can identify some of the genes which are involved in the pathway. Then you can identify some proteins which are important to drive the disease. And this is all huge amount of effort which comes from a lot of research in academic setting, lot of publications which are in the public domain, lot of data which is generated, including some of the basic science simple experiments like western blots, but also a lot of sequencing experiments, including multi-omics, like genomic sequencing, or doing proteomic metabolomics and so on. So target discovery is an essential phase of the drug discovery process, is the very first one. And to qualify for making a drug, any target needs to fulfill few criterias. Okay. It needs to be tractable. Tractable meaning that any protein which you identify as a target which could impact a particular disease it needs to be tractable by something, either a small molecule drug or an antibody or a protac or antisense oligonucleotide. There needs to be some way that you can actually target that protein or that particular gene. It is most important thing in the drug discovery process, because if you cannot make it tractable or you don't have any tools to make a small molecule or antibody, you will never be able to target a disease or that particular gene or protein of interest. There are other ways how people do this, and it is also with high throughput sequencing, high throughput screening. For example, in the big pharmaceutical company, if you know about a particular target, they have a lot of drugs which are already in the library, and they use those drugs in a functional assay to screen which drug is most effective against that particular protein. And then you can choose that drug from the screen and develop it further in the drug discovery pipeline. There are structural biologists and computational chemists who are essential, and they are mainly playing an important role to modify the drug. Now imagine you have a protein and then you want to design a drug. It's like a perfect lock and key type of mechanism. But when you actually have the high throughput screening or some drug coming from the library, it might not be like a perfect fit, but you want to make it a perfect fit. And that can be done with the help of structural biologists and people working in medicinal chemistry and computational chemistry, who can modify the structure of the small drug and make it a perfect fit for the protein. Or you can have an antibody, and then people can design they can modify some of the epitopes, um, and they can modify the variable chains of the antibody, which can recognize the different epitopes which are either coming from foreign particles, either viruses, bacteria, or against a particular protein of interest. There are people who are medicinal chemists, and they are specifically playing an important role. Once you have this lock and key mechanism, some of the final modification needs to be done, and which is mainly, for example, if you have a small molecule drug, it binds to the protein, but you want now that this is like an agonist or inverse agonist, and those small molecules needs like some modification, and they are mainly taken care by medicinal chemist people within the pharma company. Once you have a molecule, then the next step in the drug discovery process is around pharmacology and toxicology of the drug, which means doing all the experiments which are needed in a preclinical space. So, for example, giving the drug to the mice, looking for how much is the absorption, metabolism of the drug, where it is deposited, whether it is getting cleared or not, whether it has any toxicity issues or not. And then at the same time, running these experiments in the mouse models. So for example, if you have a drug against obesity, you would like to run this in some of the mouse experiments, which is mainly for obesity mouse models, and then try to see whether your drug is having any effect or not. And this is extremely important, because if there is any toxicity issues, or if there is any off-target effects, or it is affecting some of the other organs, or it is not showing you the half-life which you are expecting, the program actually gets terminated over there. Next important step is, once you have the drug and you have already done some pharmacology and toxicology testing, Galnix is another important phase of the drug development, where there is a team of people who are actually looking for how best you can store the drug so that it has the higher half-life and it does not affect its property. So for example, if you go to a pharmacy, you find some of the drugs are in a form of syrup, or there are some medicine which are in the form of gels. There might be some in the form of tablets, or some in the form of capsule. And this is all decided by actually people working in Galnix, because they are the one who are looking for the stability of the drug over a period of time, and what is the best way that this drug could be made available for the people who need the most. Once you pass through all these phases, then it starts with the clinical development phase, which is phase 1. This is first time any drug is actually given to the patients. And usually this is in healthy donors. So there are people who are healthy and they will be taking the drug. And the main aim of this experiment is to look for any toxic side effect, to look for pharmacokinetics and dynamics of the drug, how much it is getting absorbed, how much is the half-life, is there any toxicity issue, how well the drug is tolerated, how much dose needs to be given to these patients. If it passes the phase one, it goes to the phase two and three, which is mainly big clinical trials. And these clinical trials are done on patients who have a particular disease. So, for example, you must have seen during the COVID time that companies were doing the phase two and phase three clinical trial on COVID patients and patients who are not having COVID. And these are usually multi-central trials and in multiple different geographical locations. And if you get success in the phase two and three of clinical development, then the next what you do is you apply the knowledge um, from the clinical trials to pharmacogenomics. And this is a kind of new field. But the whole idea is that if you have a drug, you want to make sure that where your drug will work the most. Meaning, if you take a drug and it works in one patient, it doesn't mean it will work in every single patient. And this is mainly because we all are slightly different in our genetic material, and there might be some mutations which are in our genome, and that might have effect or different effect of taking a particular drug. So the whole idea is that you are giving a drug to a population or to a particular individual, and you are making sure that this patient will respond, and maybe this patient will not respond because they have a particular mutation. So this is the whole field where people are working, and this is also the concept of precision medicine, because based on some of the mutations or some of the biomarkers, you can actually predict whether a particular patient will respond to a given treatment or not. After all this, the information which we have collated right from the drug discovery phase to the phase one, phase two, phase three clinical trials and pharmacogenomics, everything is compiled together. And this goes to the approval agencies like FDA, for example, and eventually the drug is approved. So just by looking at the whole process, one can actually appreciate the fact that it is not one type of professionals who are actually doing this. It's not only the scientists. We have scientists, engineers, structural biologists, chemists, pharmacy people doing the pharmacology, toxicology, doctors, nurses. Lots and lots of people are actually involved throughout this process. And usually it takes 10 to 12 years from the time you have actually identified a particular target and it gets approved by the regulatory bodies and it comes to the market. So it's 10 to 12 years of total time. Now, the question is that this has been happening traditionally. So this is how the drugs are developed, and still they are developed in exactly the same manner. But how we can actually accelerate each of those phases. So for example, how we can identify the targets much more faster, how we can do the pharmacology or toxicology studies much more faster, how we can identify which patients we are going to choose in the phase one, phase three, or phase two clinical trials, how we are going to choose those patients who are having, say, a disease, say rheumatoid arthritis, and just making sure that our treatment will actually work in those patients. So there are n numbers of these questions, and these questions are not a simple questions to be answered. And that's why the field like multiomics is super important, because biology is extremely, extremely complex. And having a robust understanding of the disease, the biological pathways and mechanism can actually help us to accelerate the whole process of drug development. And hopefully, with the use and integration of these technologies, we will be able to develop drugs at much faster pace. So what is multiomics? Multiomics, as the name itself suggests, it's multi, meaning there are multiple different technologies. And then it is omics, meaning it is an untargeted approach. So I'm sure most of you have performed some qPCRs or normal PCRs, and I'm sure most of you have performed, say, Western blots, or you have looked at some of the specific mutation by doing some simple PCR. Now imagine if you can actually do this at scale in an untargeted manner, meaning you can do qPCR, or you can look at the gene expression of 30,000 genes across the genome. That is transcriptomics, meaning you are looking at the transcript, but at the whole genome level. So omics is actually looking at the different, um, I will say, different layers of information in an untargeted manner to have the maximum confidence about a particular disease or a particular pathway or a particular cell types, and then to have take much better informed decision to do your downstream analysis. So for example, here what you can see is we have DNA, and this is central dogma of life. So DNA can be replicated, and you basically have DNA. DNA can be transcribed into RNA, and then you have protein. Ultimately, what really matters is that you have protein, which is a functional protein, because that's what is going to have a physiological effect on the cell, and eventually will show some physiological effect at an organ level or at an individual level. Now, if we take those three different segments, for example, DNA, RNA, and protein, we can actually categorize different omics technologies. For example, genomics or whole exome sequencing, or epigenome, meaning looking at the DNA methylation or chromatin accessibility, histone modification. If we look at RNA, meaning we can do some transcriptomics, we can look at some mRNAs, microRNAs, tRNAs, long non-coding RNAs, and we can look at proteins by looking at the whole protein, which are expressed in a cell or in a health versus disease setting. We can look at specific protein modifications, for example, phosphoproteins, or we can look at metabolites, which are present in the cell or at a tissue level. So if we have all this information, one of the biggest thing and the biggest advantage what we have is we are not limiting ourselves to a one particular gene or two particular gene or one particular pathway. Rather, we have huge amount of information which is at different layers, meaning you have DNA, RNA, and protein, and we can filter all those information with the high performance computing and with a lot of computational programs which are being developed. And then we can apply this to multiple different fields. For example, identifying different cell types, looking into tissue and tumor heterogeneity, or generating atlas. Atlas is like, we have, you can consider this as a map, which is like a world map. But in this case, it is an organ map, meaning in a particular organ, how these cells are arranged in space. So you can look at the architecture of the tissue, and at the same time, you can also look at what are the different cell types which are present in the tissue. It could be used for the biomarker discovery, meaning can we have some predictive biomarker? For example, someone has cancer. Can you predict whether a family member in the same family will have cancer or not? So there are some of the predictive biomarkers which people can use. There are some companion diagnostic biomarkers, meaning that if someone has been given a drug, whether that person will respond or not respond, you can actually predict that. So it's an essential and very important field of in the drug development process. You can get insights into complex disease, novel pathways, and network. And this integrated approach actually provides much deeper understanding of disease mechanism and potential therapeutic targets. So far, I've just explained you the overall concept of the drug development pipeline, which is quite extensive and can take 10 to 12 years. And then there is a multi-omics, which is having information, huge amount of information in an untargeted manner to have the layers of information from a particular disease, tissue, or cell types, which are treated or untreated, diseased or healthy, and then use those information to impact patients or to impact the drug development process. Now, I just want to give you a couple of cases, which are mainly from the publications. not my publication, but just publicly available data sets, where they have nicely shown that how multiomic technology could be actually used to some of these things which are mentioned here. So, for example, patient stratification or looking at some of the biomarkers. So, the very first example I want to give is of idiopathic pulmonary fibrosis. It's a lung disease. It's a chronic progressive interstitial lung disease, and usually it starts at the base and at the periphery of the lung. So we have lung, and it starts actually from the outer side of the lung and at the base of the lung. And this can be present in sporadic form or the familial form, meaning you can have some familial history, some mutation in the genes, and people might be at risk of developing IPF. Or it could be just a random thing and can happen to anyone. And it is usually associated with increase in cough or shortness of breath, with a devastating effect on patients' quality of life. And you can actually relate this because patients who actually have IPF, they have difficulty in breathing. So something similar to what we observe also in COVID infection. A lot of research has been done which shows that this is an epithelial driven disease, meaning that the epithelial cells in the lung are the most important drivers of the disease. And what they do is they actually produce a lot of mediators of fibroblast migration, proliferation, and differentiation, which results in a myofibroblast formation and tissue stiffness. We do have drug against this particular disease, a couple of drugs which are antifibrotic drugs. But one of the sad thing is that these drugs are effective only for up to 24 months. So imagine if someone has IPF and they are on these two drugs, they can actually survive just for 2 years. And that's it. That's the best treatment what we have for these patients. And the common side effect of these drugs actually include diarrhea and nausea. So this one slide already tells you a lot about the disease. It's a complex disease. We don't have drugs. The best drugs which are available are just for 2 years of patient survival. So clearly, we do not understand the disease really well. And because we don't understand the disease really well, we also are not able to develop efficient treatment against this particular disease. Okay. So what we can do applying the multiomics to understand the disease and to enable the whole process of drug development is we can try to study this in a little bit more detail, and we can use those detailed information to inform us about some patient stratification. So this is just a pictorial representation of what IPF looks like. So for example, what you can see here on the left hand side is the IPF lung, and this is this is the healthy lung, and this is the IPF lung on the right hand side. And what you can see is in a normal condition, you do have your healthy lungs, and this is the area which is dark brown in color is your bronchioles. And then you have your alveolus, which is super nice and round shape. And this is the place where there is exchange of oxygen and CO2, which is taking place. So you have your alveolus here, and you have your blood vessels here. And this is where, when we actually inhale, so when we take when we breathe, we are taking the oxygen. This is where the oxygen and CO2 exchange is exactly happening in the lung at the interface of alveolus and the blood vessel. But what happens in the case of idiopathic pulmonary fibrosis is you can see the shape of these alveolus here. They become like capsule-like structure. So that's one thing. But at the same time, what you see is there is a lot of matrix deposition, which is shown in the lines, which are basically the fibrocytes or fibrosis with these lines shown here. So usually this is absent in the case of healthy individual, but when there is a fibrosis, you actually have a lot of fibrocytes, a lot of extracellular matrix. And as a result of that, this is what is happening. So you have the fibroblast or fibrosis here. So you have a lot of fibrocytes, and these are myofibroblast cells. And what is happening is this space is actually getting much more broadened. So the capacity by which you can actually exchange oxygen to CO2 is reduced massively, and this is the reason why these patients are not able to breathe properly. If we look into the bit of biology of the disease, what you can appreciate from this figure here is you have a healthy alveolus, and these are alveolar epithelial cells. There are two types of alveolar epithelial cells, which you can see here. So this is one type, and this is the type 2 type of alveolar cells. And if there is some epigenetic programming, or there are some repeated micro injuries, or if there are some genetic susceptibility, including aging, you can actually disrupt your epithelial layer, which you can see in this figure. And this could be multiple things. So there can be epithelial activation, or there can be epithelial cells to mesenchymal cell transition. And it can be a senescence, or it can be epithelial apoptosis or proliferation. So multiple factors can play a role. This results in increased secretion of growth factors or matrix metalloproteins, some of the developmental signals. And as a result, what happens is you have lots and lots of fibrocytes, or fibroblast deposition, myofibroblast tissue becomes really stiff, and the exchange of O2 and CO2 is really hampered in these tissues. This is really bad condition, but we can study a lot if we apply multi-omics from the patient tissue sample. So this figure is just a quick overview of the study. So what you can see here is, in this study, people have taken the control samples and the IPF patient lung tissue. So this is actual lung tissue taken with the help of um, it's a small biopsy or a small piece of tissue taken from the lung of control and IPF patients. And what you can see in the image here is that you have a nice tissue which looks really good, and then you have the IPF on the other side. You can do an H&E type of staining, which is a histopathological staining, which is done routinely in the clinic. But what we can do is we can nowadays apply some technologies or tools which can help us to identify the gene expression at a tissue level without disturbing the tissue architecture. So basically, you can take the tissue and you can perform the Visium technology. So this is 10x Visium. And for now, we will not go into the details of technology, but just imagine that you have a tissue which is on the slide. And within the tissue section, you have different cells which are present here. Now imagine you can do the amplification of genes without disturbing the whole tissue. So you are actually doing amplification at a single cell level throughout the tissue. So just as an analogy, imagine if you're doing a qPCR, usually you put like a small tube and then you look for a particular gene, you amplify the gene and you get the expression. But with this technology, you can have the whole tissue and you can literally amplify genes present in every single cell in the whole tissue. And this is what you see here in this representation. Every single dot which you see is a single cell, and every single color which you are seeing is a different types of cells. Okay, so we'll go into a bit more detail in the next slide. But for now, this is a technology which is Visium technology, and it can look into the gene expression in an untargeted manner, meaning it will amplify exactly as, say, someone is doing a bulk transcriptomic. It includes every single gene it can detect. There will be some exclusion, like low expressing genes might not be detectable by this technology. But to complement a Visium technology, there is another technology which is known as Xenium technology. This technology is also from the 10x Genomics, is the same company. And what you can do is Now you can go to a single cell resolution in the tissue. So imagine every single cell as a test tube, and then you can actually amplify the mRNA of your interest, and you can amplify up to 5,000 genes or 5,100 genes using a Xenium panel, which is literally like running a microarray type of experiment or running 5,000 different qPCRs at the same time. So this technology is untargeted. This is a targeted technology. So you can do minimum number of genes. And then there is this technology, which is known as COMET. And what you can do is you can actually look at the protein expression in space. So without affecting the tissue architecture, you can look at the protein expression. So just to quickly summarize this slide, imagine you have a tissue, and in the tissue, you can look at individual cell types. You can look at the gene expression in individual cell type, and you can also look at the protein expression in every single cell type without disturbing the tissue. So that is literally like a lot of information, which is different types of cells, different tissue coming from different locations, and so on. At the same time, what we can do is we can use some of the single cell sequencing datasets, which are either generated in the study or they are coming from a publicly available dataset. And the difference between this technology and this technology is here we have not disturbed the architecture of the tissue. Okay, so the whole tissue is still intact. Here we have taken the tissue, we have dissociated the tissue into single cell, and then we are looking into the gene expression of a single cell. And the power of this technology is we have histology, we have transcriptomic, and then we have proteomic, and we can overlay the whole information in one slide so that we can now look at the tissue which was inflamed. We can check what are the cell types which are present in the inflamed region, what genes are present in the inflamed region, rather than looking at the whole tissue. Okay. So once we actually run all these technologies, you have to do a lot of data analysis, which is high performance computing, but it can give you a lot of information around pathology, around which cells are interacting with other cells, what is a receptor ligand type of relationship, what are the specific niche within a tissue, meaning you can have some areas in the tissue which is having a lot of T cells, or some areas having a lot of B cells, or some areas having a lot of fibroblasts, because you can literally see the whole tissue without disturbing the architecture. You have lots and lots of information which you can process. So now let's see, if we do all this, what we get in return and how we can understand the disease pathology in a better way. So for example, in this figure, what you can see is you have control. So this is the control tissue on the top. So this is the control tissue. And at the bottom panel, you have a tissue coming from the IPF patients. Okay. Now focus on the top panel here. So you have 81 cell types. So these are alveolar type 1 epithelial cells. And what you can see is lots and lots of blue color. Okay. So this is the scale. And you see a lot of blue color in the control tissue, meaning if you are taking a healthy lung, you do expect lots and lots of alveolar type 1 epithelial cells present. But if you look at IPF patient here, what you can see is there is striking difference in the number of 81 cell types. So you don't see epithelial cells. Most of your epithelial cells are completely gone. So one of the very first thing with this technology, what you can observe is that if you take a tissue, A1 type, alveolar type 1 epithelial cells are diminished in the IPF patient in comparison to control. Now, the next thing which I mentioned was around the pathology of the disease. And one of the key pathology is, as the name itself suggests, it's idiopathic pulmonary fibrosis, meaning it is a lot of fibrosis. Fibrosis is an, you can say, result or outcome of a lot of myofibroblast. If you look at the control tissue, you don't have myofibroblast. So in a healthy condition, in a healthy lung, usually you won't find myofibroblast. If there is an injury, usually there will be some myofibroblast for some time during the tissue repair, but later on they will just die, and the tissue becomes normal. But if you look at this and then compare the IPF lung, what you can see is there is lots and lots of myofibroblasts which are getting accumulated in the lung tissue. There are some patches of the lung where you have lot of myofibroblast deposition. There are some patches where there is a mild deposition. There are some patches which are just not affected. And this tells you that actually disease is progressing from the periphery. So what you can see is this is the periphery of the tissue, and you can see the maximum blue color, which is at the periphery. And then slowly as the disease progresses, you actually have more and more myofibroblast and more of a fibrotic tissue, which results in a very severe form of IPF, and eventually these patients die. Now, the next thing which I want to show is the macrophages, which are FAP-P4 positive. So what you can see in the figure is a lot of blue color, which is suggestive of the macrophages, which are FAP-P4 positive. And if you look at IPF tissue, then these macrophages are much more diminished in the number in comparison to control. So this is then spatial information, and it clearly tells you about what cell types are getting changed in health versus disease setting. Now, if we go into a little bit more detail and do a little bit more advanced computational analysis, we can actually look at the areas within the tissue which are different or common. Okay, so I will just walk you through this heat map. What you can see is on the y-axis is different niche. And niche meaning you have a tissue and the whole tissue is not homogeneous, meaning you will have some places of the tissue which is having more of an alveolar cells or more of immune cells or more of airway cells or more of fibroblast cells. And this way you can actually categorize, rather than taking the whole tissue, you can categorize it into small pieces. Okay. So these are different niche, or you can say different areas which are present in the lung tissue. And on the x-axis, you have different cell types which are either present in a very high number or present in a low number. Okay, so these are the areas, or if you just imagine as an analogy, think of a Google map. So you have the Google map and you have different areas of the city, and within the different areas of the city you have different shops. Okay, so imagine that for the tissue you have different areas and you have different cell types. What
Excuse
you
me,
can
sir.
see in this map is there are red colors which are super red, meaning these are the areas which are representative of these cells which are maximum in number. So for example, this is alveolar niche, and alveolar niche is characterized by alveolar type 2 epithelial cells or capillary eriocytes. Similarly, if we talk about the fibrotic niche, then these fibrotic niche are basically characterized by the myofibroblast population, which I've just showed you. If you look at the macrophage niche, then it is colonized by different types of macrophages. Just to see this in a very simple format, focus here on the colors. What you can see is in the control, you have lots and lots of orange color, which are basically your alveolar epithelial cells. In the disease setting, you can see the orange bit is decreased significantly, suggesting that alveolar niche is significantly decreased in IPF patients. If you look at the red color here, you actually don't have any red color because healthy tissue don't have fibrosis. But in a fibrotic condition, in an IPF condition, you have lots and lots of cells which are basically fibrotic niche, meaning these are the areas in the tissue which are having lots and lots of myofibroblast, which is also represented here in this heat map. So with this, what you can appreciate is we can look at the lung tissue, we can look into the areas of the lung which are different, we can categorize that area into different niche or different areas of a Google map, and then we can identify which cell types are playing the most important role, what are the genes and pathways which are upregulated or downregulated, and are there anything which could be targeted for the drug development process. At the same time, because we have the tissue level information, now we can categorize the patient based on how much fibrosis they have. So we can look into the patient and say like, okay, this is an early phase disease, this is the late phase, and maybe this is something which is much more terminal. Or we can say, because there is a lot of fibrosis, we can give them this drug. Because there is less fibrosis, maybe we just need to give some immunosuppressant to these patients, and we don't need to give antifibrotic agent. So this information is extremely crucial if you think from a patient perspective, because if we can have this information before, we can give right patients right treatment. We can categorize these patients based on the type of disease they have, not IPF, but within the IPF, what is the category of the disease in which these patients fall. Now, I just want to give a quick example of rheumatoid arthritis, which is another autoimmune disease. It is inflammation of the joints, and usually it is with systemic comorbidities driven by the breach of immune tolerance. The good thing is we have almost 60% of the patient who basically have long lasting remission, meaning somehow you can suppress the disease. They still have a disease, but they don't have any flares, or the disease don't come again. But roughly 40% of the RA patients still do not achieve long-lasting disease remission or restored state of immunological homeostasis. Meaning, even today, we have lots of drug for rheumatoid arthritis, but 40% of those patients, meaning 40 out of 100 patients, do not have long-lasting remission. Drug-free remission is achieved in 10 to 20% of RA patient, meaning that if someone has RA, rheumatoid arthritis, and they don't take a drug, the chances that they will again develop rheumatoid arthritis is almost 80%. Meaning we still are not in a position to actually develop a drug which can provide an immunological cure, or we are not in a position where if we remove the drug, the patient, they don't need drug anymore. So they have to take the drug in order to keep the disease in control, and so that it doesn't progress and doesn't become much more pathologic. Just as an overview, what you can see here is the joint. And in the joint, usually it is quite sterile overall. But as the disease progresses, or if there is some susceptibility for RA, the preclinical RA is something like, you can see there is a single cell, which is like an immune cell, which infiltrates in the joint. And because there is immune cells and they are releasing these cytokines, it is attracting a lot more immune cells to the joint. And you can see there are lots and lots of immune cells which are in the joint. And because there are lots of immune cells in the joint, there is more inflammation, more immune infiltration, and that results in the hyperplasia of the lining layer. And it also results in a lot of cytokine release, mostly the inflammatory cytokines. And the question is that what we can actually do to either predict what is the disease progression, or can we use the omics technology to actually predict the response, whether someone will develop a flare or reoccurrence of the disease in future or not. So this is a paper which was just published, I think a few days ago. And they have used the multi-omic technology, mainly the single cell technology, to investigate what are the drivers of the disease, and then look for the biomarkers which could be predictive of flares. So what you can see here in this immunofluorescence image is this is healthy synovium. And what you can see beautifully is the red line. So the red line is here, which is like a thin lining. And then you have the dendritic cells, which are shown in green color, and blue color is the nucleus. So if I just go back to this figure, remember that this lining layer, which you can see here, is a thin lining. Healthy condition, you have this thin lining, which is here shown in the red color, and then you have a lot of immune cells which are here, which are like DCs within the tissue. If there is an active RA or active rheumatoid arthritis, what is happening is this lining is completely disturbed. So you can see that now red cells are kind of like everywhere, and that structure of the lining is disturbed, and you have more and more of DCs, which are dendritic cells, getting infiltrated in the tissue. Now, once these patients are actually treated with some drugs, they are in remission, meaning they don't have any disease or they don't have active disease as long as they are taking the drug. And in the remission phase, you again have this very nice lining of the red cells. And then you have the green cells, which are your dendritic cells, but they are much less in number in comparison to the active RA. So what authors have done is they have used the single cell technology. So this is single cell RNA sequencing, meaning they have taken the synovium biopsy, dissociated the biopsy, performed the transcriptomics, and these are the markers which they have used to identify different dendritic cell populations. So these are dendritic cell 1, type 2 cells, dendritic cells type 3, or dendritic cells type 3, which are IDCs. And what they were able to show beautifully in the FACS experiment is that if you have active RA and you gate on CD1C, which is a dendritic cell marker, you can see the population over here. So these are the cells which are CD32, CD39 positive, and these are CD1C positive cells. You have these cells which are super increased in an active RA, but once you give the drug, you actually decrease these cells in number, and you have more of the cells which are CDC1 positive but not positive for CD14. So this is just a validation of what you can get from the omics dataset and then validate on the facts. But most interesting thing is they can actually use the patient blood. So for example, you can take the patients who are under remission. So these are RA patients, and they have been given drugs which are basically the drugs which are rheumatoid drugs, and they have taken the peripheral blood from the patients, and they take these bloods after 6 months of the treatment, or they take at the disease flare or after 6 months of the disease. And they are using the blood samples to perform the BD Rhapsody, which is a single cell technology, but more a targeted technology. So you can have genes which are specifically amplified, which can be your target of interest. It's not an untargeted technology. It is something which is targeted, meaning you can choose, say, 100, 200, 300, or 400 genes of interest, and then you can look at the gene expression at a single cell level. This heat map here, it just shows you different population of monocytes. So you have this population here, which is red color. So these are monocytes. You have this purple color, which is DC2, or you have yellow color, DC3. And these are the genes which are the marker for those different cell types. Now, if we perform this multi-omic type experiment, we can actually now look into what are the genes which are differentially expressed in the patients who are in remission, patients who are in drug-free remission, meaning that these patients, they are not taking any drug and still they are in remission. Remission which is maintained, meaning these patients do not have active disease, but they do take the drug. These are the patients who are in remission, so they didn't had any disease, but later on they develop the disease. And then these are the patients who actually have an active disease. So we can take all those 4 different groups and then look for the differential gene expressions which are associated with each of those groups. And we can use this information. So you can see these are actually the genes which come from this analysis. So these are 24 set of genes in different groups. And now we can use this information to predict whether a patient will develop a disease or they won't develop a disease. So now focus on this one. So here, this is the IDC3 population. So this one in the red color. If there is a remission at the baseline, you can see these cells are kind of few in number. But if there is a disease, then it all becomes red in color, meaning IDC3 population, which is the dendritic cell 3 population, they increase. If there is a drug-free remission, this population is much less. If there is a point of flare, then again you have a lot of red color, which means that if you are actually looking into the RA patient and you take the peripheral blood and just quantify the number of IDC3s, meaning the dendritic cells, you can actually predict whether the patient is having an active flare in the joint or not. And this is an interesting analysis where they have looked at week 8. So you can see everything here is in blue color, meaning all these genes are super low expressed. And as you come close to the flare, meaning the patient is not having any disease, and then 1 week later, what is the gene expression? 1 week later, what is the gene expression? 1 week later, what is the gene expression? So you have -8 week, -7, -6, -5, -4, -3, 2, 1, and 0. And this is the point when you actually have flare, meaning there is an active RA disease. You can already see from almost minimum gene expression or no gene expression of these set of genes, you have these red colors which keep on popping up. And at week 4, you can already predict whether a patient will develop a flare or not. So this was possible because we can actually use the multi-omic technology, not because we can do a single gene PCR, because nowadays the data is increasing in the volume. And with the increased amount of data, it is almost close to impossible for human eyes to predict those patterns. So based on all this, what I mentioned, where we are moving forward with multi-omics. Multi-omic is a field which is extremely moving with an unprecedented scale in different areas. For example, I mentioned you about spatial cell spatio-temporal profiling, meaning you can look at the cells in the space and with the time. So I just showed you example of RA, where you can take the sample, the blood sample at 1 week, 2 week, 3 week, 4 week, and then you can predict what are the gene expression changes. All these information which is generated, they need to be integrated in a manner so that we can make meaningful conclusions. So for example, having the proteomic data, metabolomic data, genomic data, and then integrating the whole data set to make meaningful patterns is around multi-omic integration. Once we have integrated all these data set, we can make use of the information. As I just showed you that in RA patient, you can predict whether a patient will respond or not. This field is known as precision medicine. You can then do some functional validation like a FACS experiment. So you can run the FACS experiment to validate the findings from multi-omic experiment. And at the same time, you can develop some machine learning algorithms, which are extremely important in identifying the patterns in the data set. So what you can see here is the neuronal network. So if you have all these big data sets with hundreds and thousands and millions of parameters, human eyes are not able to detect those patterns, but with the machine learning tools, now you can identify which cell type is associated with a disease or which drug is associated with a response, who are the patients who are responding or non-responding. This field is still evolving, but there is a lot of promise which it has already shown, and a lot of people across the globe are continuing to advance this technology for the drug development process. In summary, I've showed you that multi-omics actually holds significant potential for transforming disease treatment and improving the patient survival. It enables the development of personalized medicine and more effective drug discovery. And what is really needed is the global collaboration and in-depth research, which is important because if there are few patients on which a particular group of scientists are studying those patients, we need to have all these different small groups combined together to have the huge amount of information which could be collectively used to advance the multiomic research and help the big pharma companies to actually aid in impacting at scale. Usually, this is possible if there are academic collaboration and industry collaboration coming together, because it is extremely difficult for the big companies to have access to the patient sample. But on the flip side, academic institution hospitals might have more samples, and having a collaboration or a global collaboration can actually enhance the whole process of multiomic drug development. So lastly, if you are actually interested in what I spoke today around multiomics and translational research, follow me on LinkedIn. And I also write some of the blog posts, which is the Science Circuit. And this is in a very simple language for anyone who is interested and passionate about translational research, multiomics, or data science. With this, happy to take any questions.